Oh my goodness, here comes the shrimp.
So for those of you that are just joining us, this is 1972 part 3 of Attack of the Giant Brine Shrimp. And we thought it would be a really fun way to kind of start this show about microbialites and brine shrimp. Hey, Bonnie, does that look like a brine shrimp to you?
Yeah, it looks a little different. You'll have to count its penises because I understand that it has 2 penises.
Well, I was going to ask you if you think this is a female or a male brine shrimp.
Well, I don't see the claspers on the front, but
and
maybe.
I don't see a brood sac.
I don't see a brood sac. In fact, the anatomy is way off. I would say that no one actually consulted with anyone that knew anything about brine shrimp for this. And I would have done it for like.
It could just be the difficulties of the medium. It's not easy to work with clay.
Yeah.
In extreme anatomical accuracy.
That's a good point. I do see Brigham Young being carried off. And Neville says, not Brigham Young, no. She says, you didn't show Saltaire.
Okay, so I wanted to show in part 1 is the Saltaire version, but it has young people that are making out, and I decided that it made me uncomfortable watching that with everybody else. And so this is why I chose this one.
Lots of tongue, says Anne. Yeah, but talking about
sounds a little more than PG.
Talking about double penises on a brine shrimp doesn't bother you, however.
I think this video is really kind of it's like an allegorical warning. This is why we want to drain the lake over time, because this is what happens when it gets too high, is the brine shrimp get out into the city.
Maybe it's going to happen if they lose their habitat, and then they don't have anywhere else to swim, and so they're like, all right, we're going to become land creatures now.
Oh yeah.
Is it a metaphor about ecological balance?
Ooh, that's heavy. It's all Kennecott's fault. They haven't seen it. It's one of our chat. Oh, nice. I don't even know where they got these mouth parts. They look much more like the brine fly larva.
Oh yeah, they do. Those brine flies, they kind of like crunch and munch along those microbiolites.
Uh-huh.
I wondered if this is how brine shrimp might feed on the microbiolites. Maybe somebody else knows that.
Where's Melody? She watched a lot of microbiolites.
Yeah, as they crawl up the sides, kind of. I have a picture of that in my presentation that kind of looks like that, where they're like right next to it. So could be if they're surrounded by water.
I think, is this the scene where Robin's minstrels get eaten?
There's better music.
Do brine shrimp make noise? Because I've never heard them say anything like this.
Oh, you're listening to them, Matt, in the lab?
I hear nothing. They're very quiet.
I mean, I've only listened to them from above the surface of the water, so.
Anne is asking that we add the sound, Jaimi.
Is that better, Anne?
Yeah. So dramatic, she says. It's all the way to the top of Moroni.
I did, when I was counting a lot of brine shrimp, I did have nightmares about their eyes, because they're little beady black eyes. I would just imagine them looking at me, and I had lots of bad dreams about them.
Is that a giant bird now?
That would be a seagull.
That's our state bird.
It doesn't look giant enough.
Is it gonna? Oh, it's running off with it.
Yeah, and Matthew doesn't know this mythology, but see, the gulls, they actually saved the early settlers because they saved them. They came and ate all of the crickets that was plaguing their early crops. So gulls have this like very important mythology in Utah culture. That's where that whole thing comes from. Do you think that's enough?
That's good. I think that's a good start.
Welcome. This is the 6th Salty Science seminar in our Salty Science seminar series. And we're here today with a really great bunch of people. I'm really excited. My first love at Great Salt Lake was not pelicans, but brine shrimp. And so I'm looking, forward to sharing with you. Before we go too far, I'm gonna backtrack just a little bit. So remember that we have 2 ways to interact with all of the panelists. If you have a question, look for your Q&A box. If you have more comments or you want to tell us to quit playing silly 1970s cult classics, you can do that through the chat box. And we definitely welcome chatter from you all today. Wally Gwynn gave us a really great idea last time. This QR code links you to links and resources that we have given to you or that our speakers have shared with you. You can go here and find any link that we have had during during these 6 Salty Science seminars. I keep hounding you about our Great Salt Lake Institute YouTube page. There is also a QR code that's going to take you straight to our YouTube channel. And I want you to pay particular attention. My friend Alex Popora at the Utah Society for Environmental Education, she taught me about playlists. So if you click on playlists, you'll be able to find information about specific topics. Maybe you want to see what our students are up to. You might want to find the Salty Science seminar series. You might want to know, I did link to the brine shrimp folder has links for Attack of the Giant Brine Shrimp, so you can show all of your family and friends. So please use that. We're excited that people are subscribing and that they're using this as a resource. And just a little backtrack, we've gone over so much information. This is the 6th 2-hour seminar where we've just taken a headfirst dive into the salty waters of Great Salt Lake. And sometimes Dave Parrott ended up on the shoreline because he likes plants for some reason. We've talked about water and humans at the lake, definitely have mentioned birds 8 million times. We've talked about reptiles, Microbiolites, and mercury issues, the plants of Great Salt Lake, both shoreline plants and invasive plants that the state of Utah spends a lot of time controlling. A couple weeks ago, we talked about, am I on Mars or Great Salt Lake? And today, obviously, we're talking about brine shrimp and microbialites. I did get some feedback that people wanted to hear about dust at Great Salt Lake. And so on March 4th, mark your calendars. I will send you a link for this webinar once we get this recording up. But we will be talking about how to recreate at the lake, what happens, to the very important dust. And I also thought we should maybe take a step back and really explore the watershed of Great Salt Lake because it's so unique, and how does our watershed work? So please mark your calendars for March 4th. We do have a couple of really cool things going on. Confluence is going to be opening on February 18th. This is the Utah Museum of Fine Arts will have this exhibit. And a description is, where does water in Salt Lake Valley come from? Where does it end? In Confluence, the Utah Museum of Fine Arts, in collaboration with 6 local organizations, explores this essential component of our daily life. From mountain streams to Great Salt Lake, the exhibition illuminates the idea of water environmentally, culturally, and historically. And we have some really cool water columns that we put in there. We actually took water and brine shrimp into the Museum of Fine Arts, and that never, I mean, that's not ever supposed to happen. So we're pretty excited about that. The Natural History Museum, they have an exhibit called Decisions Downstream that runs January 15th through July 31st of 2021. All of us, people, fish, and many other creatures depend on the water in Utah's rivers. The choices we make about how to develop water resources have big impacts on river habitats. In Decisions Downstream, a watershed scientist teams up with artists to explore the way of seeing river habitats. The watershed scientist that worked with the Natural History Museum and artists is Dr. Sarah Null, and she gave a talk in our first Salty seminar about consumptive water uses in Great Salt Lake. So I thought you might be interested in that. February 18th, we're getting really excited. There's a QR code you can link to a live watch party. It starts at 11:30 Mountain Time and will go until we run out of things to talk about. It will be live watch party, so we'll be actually, projecting the Mars rover landing on Mars. And something really exciting that we have, I want you to link to that on that QR code. You can also go to our links and resources. But we've teamed up with the Ott Planetarium at Weber State University. And we're going to be doing some fun kind of home science things while we're there. We're going to have students looking for meteorites and then using this cool origami paper microscope that we will give to them in a kit. And we're going to be looking for those meteorites. Also, we are putting out, because brine shrimp have gone into space and we think that that's really super cool. We are starting this, we'll give out some of these beaker bags, these briny brainy beaker bags. And again, the QR code that's in the top of your screen, that's gonna link to a webinar. If you sign up for that and we have enough kits for you, we will have you pick some kits up from Westminster College. And I did bring along my friend, Matt Huber. He's from Algae Research Supplies. Are you here, Matt? Can you hear me? Can you talk?
I am, I'm here, hi.
Okay, cool. So, so Matt actually found us, and I'm really excited because I've been trying to get brine shrimp into every single classroom and like saving the lake one brine shrimp at a time. And when I met Matt, he's kind of crazy about algae, like I'm crazy about brine shrimp. And so together, I think we make a pretty awesome partnership. So do you want to talk for a sec, Matt, and tell them about your kits and what you're up to?
Sure. Hi, my name's Matt. Kind of a couple seconds about myself. When I heard it was tiaras today, sorry. So real quick, I got out of undergrad and worked in pharmaceuticals for a handful of years. And I had this really wacky boss who said, "You did a good job with this stuff, but I don't think your heart's in it." And it wasn't. So he said, "Okay, go to the desert. So go to the desert and don't come out and don't bring anything but water until you know what you want to do with your life." And I took his advice, and I realized while I was there that I was put on this planet to save the planet. So, and to do that, it was going to require microalgae and a thorough knowledge of carbon dioxide science. So I went to grad school, studied biological oceanography, and then got into growing algae cultures. So I've been all over North America, growing and building large-scale outdoor farms. I work for General Atomics and for another company called Earthrise that grows spirulina. And the biofuels industry got really, really weird. And so I took my lab and I converted it into education. And that's where we are now. And that's kind of what brought us here. So I've been growing algae and selling it online, and I noticed that there was a real need to teach the conversion from that primary producer into the primary consumer, which in this case in brine shrimp. And using brine shrimp as a model was just insane because it's easy to package, you can ship it. So I had came up with this idea called Brainy Brinies, and it took several years to get it up and off the ground. And finally, I just started reaching out to people who I thought were smarter than I was, and I found Jaimi. And immediately, she and I clicked. Our missions were so completely in the same place that it was really fun to get to know her. And then I got to know this other group out in Tacoma, who was also they're called the Foss Waterways Seaport. And we all kind of pitched in to make this kit that you see in front of us really awesome. So we've done some minor tweaks on it recently, but this is now something that we have it online for like 26 bucks. We can go as low as like 10 bucks, or if your school is at need, we'll drop it down to 5. But basically, we just want to teach algae and brine shrimp and see that trophic energy transfer and get that out there. Our mission is to have every single person in the US grow algae before they graduate.
Cool. Thank you for coming.
Sorry, that was too long.
No, I surprised Matthew and put him on the spot today. So we do have a really great question. Vicki Turner wants to know why brine shrimp aren't the state shrimp. And it's one of my goals in life. There's only 7 states that have a state crustacean, and Utah is not one of them. And so someday in my illustrious lake career, I hope to add that to the list of what we've done. So thank you. So we're looking at our Brady Brinies. I'm going to move these. They're super fun. I had 2nd graders who are in remote school today set these up. So a 2nd grader can set these up. They're very simple and very quick and easy. So today we're going to start, last week we were at the north arm of Great Salt Lake, and today we're going to be in the green south arm that's about 15% salt, about 5 times saltier than the ocean. And our first speakers that I want to bring on, I always feel really lucky that I get to meet all of these really cool, nerdy, awesome scientists. And Eric Boyd, he is definitely one of them. Eric is at the Montana State University, and we tease him that he's the big LeBoydski. We have a lot of fun joking around with each other. And so I would like to invite and it's one of those really awesome collaborations with Eric's lab in Montana State. With a lot of people, graduate students who are putting out amazing research. And so the first person that we want to bring on here is Melody. Melody Lindsay is currently a postdoctoral scientist at the Bigelow Laboratory for Ocean Sciences in East Boothbay, Maine. Oh, let me take this off. Here we go, that's better. She's developing methods to link cell-specific genome data with activities of single microbial cells from low biomass subsurface environments. She graduated with her PhD in 2019 from Montana State University, where her thesis research focused on how hydrogen from water-rock interaction can fuel microbial primary production in Yellowstone hot springs. It was during her PhD work in Dr. Eric Boyd's geomicrobiology lab that she first fell in love with the enigmatic, I can't say that, you know what I'm saying.
Sorry.
You're killing me. The enigmatic microbiolites of Great Salt Lake. She was lucky to work on several projects investigating the microbiology of microbiolites and how they contribute to the Great Salt Lake food web. She continues to be intrigued by these unique microbially built mineral structures and hopes she can see them in person again soon. And I have to say, Melody, when she sent me her bio, it's called Microbiolite. So Microbiolite, welcome. Come teach us.
All right, thank you, Jaimi, for that introduction. Okay, so yeah, thank you for having me. I'm gonna share my screen if that sounds okay. Let's see here. You get to see some really ancient microbialites as my desktop background for a little bit. Hopefully that looks okay.
It looks great.
Enough Zoominars here. Yeah. All right, so again, thank you so much, Jaimi, for that introduction. I'm here to talk to you about the enigmatic microbialites of Great Salt Lake. And so I became, or just a little bit about me. Yeah, like Jaimi said, I'm a postdoctoral researcher currently at Bigelow Laboratory for Ocean Sciences, but I first became involved with Bonnie and Jaimi through Dr. Eric Boyd's lab, and you were introduced to him a little bit at Montana State University. But I graduated in 2019. Haven't been really out west since then, but I hope to see them again one day. Maybe not during not during a pandemic. But yeah, I'm here today to talk to you about the microbialites and give you a little bit of an introduction on them, especially if you've never heard of what a microbialite is, what they are, and what do they do. So just a brief introduction on why I thought microbialites initially were the coolest things on Earth. Microbialites in the geology world represent some of the earliest evidence for life on planet Earth. And so we have in the rock record these ancient kind of layered mounded structures called microbialites, or a different term is stromatolites, that are from the Archean era. So they're about 3.5 billion years old. So these are some of the first evidence that we have for microbial mats on Earth, and they form these very distinctive macro mounded structures. This is just an artist's depiction of what these microbialites or stromatolites might have looked like on Archean Earth 3.5 billion years ago, when the atmosphere was of a different composition and the oceans. And so that's just kind of what they were theorized to look like. But we can take a look at some examples on modern day Earth of microbialites as well. So on modern day Earth, they are relatively rare. They only happen in a couple of places where the circumstances for their formation are just perfect. And so this is one of the more famous locations, and this is in Shark Bay on the western coast of Australia. But another such location for these rare microbialite structures is Great Salt Lake. And this is a picture I took back in, I think, 2015 of Great Salt Lake microbialites. And I just like how it looks compared to the artist's depiction of ancient Earth. And so if we take a closer look at modern day Earth microbialites and look at their laminated structure, we can see that there are similar patterns to what we see in the ancient rock record. So we see these different layers of microbial mats. And if we take a look at the microbialite from Great Salt Lake, this is a picture I took back in 2014, we can see that there are these layers of microbial mat atop this carbonate rock structure. So I'm going to get into that a little bit and give you a little bit of info on how microbialites are formed. And so microbialites are characterized by microbial mats. And so microbial mat forms on, in an aqueous environment. And typically in a microbial mat that results in a microbialite, this will be formed by a process by photosynthesis. And so as photosynthesis takes place in this microbial mat, the microbes will uptake CO2 and increase the pH and drive the precipitation of inorganic carbonate. And that's what lithifies these kind of domed structures. And as the mat grows, it lithifies and it gets buried. You get this concentric formation of these layered dome structures. Another thing that contributes to microbialite formation is also sediment trapping. So these microbial mats are typically, are often very sticky, and as wave action or other physical processes move around sediment and other particles, like ooids in the Great Salt Lake, for example, that sediment can become trapped in these layers of microbial mat and contribute to microbialite formation. So now I want to get into a little bit more specifically on Great Salt Lake microbialites. So this is a map of Great Salt Lake, and we can see here separated into the north arm, that's hypersaline, about 30%-ish north arm, and a relatively less saline, as Jaimi said earlier, about 15% salinity south arm. But across both of the arms of Great Salt Lake, there are microbialites, which are outlined here in this green dotted pattern. And so microbialites cover about 20% of the bottom of Great Salt Lake, and this represents the largest expanse of microbialites in the world. So despite the rarity across the world, Great Salt Lake has a lot of microbialites. And they're not just cool because of the early Earth aspect and the kind of rock record that they can inform on, but microbialite or periphyton chlorophyll levels are about 9 times that of the lake phytoplankton because they're so concentrated in these mat formations. And it has been estimated that they contribute about 30% of the productivity that free-floating phytoplankton in the lake contribute as well. So I just want to go into a little bit on the schematic of maybe how microbialites in Great Salt Lake have come into the modern era. And this is a schematic published by Mike Vandenberg from Utah Geological Survey in an article he published in 2019. And so this is what the microbialites might have looked like when they formed these layered mounds. And this is under historic conditions, so a higher lake level. And so these microbialites form these domes, but then as the lake level lowered to current day conditions, the water retreated and these domes were left exposed. And so as these domes were exposed, they kind of fell inwards, forming these kind of ring-like, or I think they're called fairy ring-like structures. And that's what we see in modern day Great Salt Lake on the shores, especially of the South Arm. This is a picture taken of microbialites off of the northern coast of Antelope Island. And you can see that they are in these kind of ringed fairy ring structures covered in a microbial mat that's kind of this brown color. So going back to this map, I'm going to tell you a little bit about a study that we published out of Eric Boyd's lab and with Bonnie and Jaimi back in 2016, where we looked at the Great Salt Lake microbialites from both the North Arm and the South Arm. So we're not quite sticking to the South Arm, we're going a little bit back to the North Arm, as Jaimi said. So I'm going to tell you a little bit more about the species that inhabit these North Arm microbialites. And here we can see they're these white domed structures surrounded by this pink water, and also of the South Arm microbialites, which are these brown colored fairy rings. And of course, this divergence in salinities of the North Arm and the South Arm, and likely what is causing differences in the microbialites that I'm going to tell you about in a second, was of course the causeway, which was constructed in 1959, which cut off river inflow to the North Arm, as many of you will know, from the Bear River, Ogden, Weber River, and the Jordan River, and it caused this great salinity divergence. So the first question we asked is what species can we look at that inhabit and specifically might even build these microbialite structures? And so we took samples, or Bonnie and Jaimi took samples from a North Arm microbialite, and if we zoom in on that, it looks like this. This is about a 2 millimeter by 2 millimeter hand sample. We can see it looks kind of salty, salt encrusted. There's not a lot of greenery, especially as compared with a microbialite from the South Arm. So if we take a close look at a microbialite from the South Arm, already just with a visual observance on this 4 millimeter by 4 millimeter sample, we can see that there are, lots of pockets associated with it, and these green spots, which are likely photosynthetic algae colonizing this microbialite, or perhaps also contributing to the formation of these microbialites. And so using next generation molecular biology techniques, we were able to quantify the amount of biomass associated with these two microbialites. And so this is a plot here on the right showing a proxy for biomass, in this case, the copy numbers of genes or DNA associated with each of these samples for both the North Arm and the South Arm. And we have the biomass that is attributed to the 3 domains of life, bacteria, archaea, and eukarya in these different colors. So on first glance, we can see that the South Arm is winning the battle here for amount of biomass. No, not really a surprise, but we've quantified it. The North Arm biomass is not really even visible on the scale, but it is still there. So if we zoom in on this area here of the plot, we can see there is biomass associated with the North Arm microbiolites, albeit at a lower level and a different scale axis than the South Arm. So again, using additional next generation DNA sequencing techniques now, we can take a closer look at the microbiolite communities of both the North Arm and the South Arm. And so this is a lot of information on the slide about the identities of species associated with microbiolites, but I'll try to take you through it, and I'll leave you with an important point about photosynthetic microorganisms. So these three plots are showing the percent community composition of different species for both the South Arm and the North Arm microbiolites for each of these three plots. And each plot represents, again, a separation of the three domains of life: archaea, bacteria, and eukarya. And so we can see that there are stark differences between the community composition of the South Arm versus the North Arm microbiolites. But what I want to draw your attention to are two species, one bacterial species and one eukaryote species. The bacteria species is this Euhalothece cyanobacteria species, which is present in the South Arm, but not present in the North Arm. And the eukaryote species is this Navicula algae species present in large quantities or large percentages in the South Arm, but not in the North Arm. And so these two species present in the South Arm, but not the North Arm, are both photosynthetic. And so this lower biomass and a lack of photosynthetic primary producers in the North Arm is likely due to the restraints caused by increased salinity. And we can see that based on this study. That was one study that we were focused on, but we also wanted to know at what point does a shift in salinity begin to affect microbiolites, their primary productivity, and perhaps other aspects of Great Salt Lake food web. So far we have looked at two salinities. And so we have the South Arm salinity of around 10 to 15%, depending on the time of year and seasons and stuff like that. And then a salinity of 30% for the North Arm. But the question now becomes, is there an ecological tipping point for microbiolite associated biomass or the food web that they likely support in Great Salt Lake? So in 2019, we published a study based on lab-based microcosm experiments where we incubated microbiolites at 6 different salinities, now ranging from 8 through 30%, in the ones you see here. And so using this experimental approach, we tracked their productivity, changes in microbial species, and then also downstream effects on some secondary consumers that I'll get into in a little bit. So just some quick results from that study in the microbiolite response to changing salinities. So in this case, we focused only on South Arm microbiolites and these fairy ring structures we see here. I've just shown this picture again for reference. And we used that in our incubation experiments to test these different salinities. This plot is a plot of the amount of carbon uptake by microbes living in a sample of South Arm microbiolite that we incubated at each of these different salinities. And so we can see that at salinities of around 8 to 15%, and 15% is the native salinity at which we sampled these microbiolites, we have relatively high carbon uptake and therefore primary productivity. However, at salinities at 20% and above, we begin to see this amount of carbon uptake decrease. So this leads us to the conclusion that salinities over 15% cause shifts in primary productivity and community composition, which I did not show that data, but that's in their 2019 study. And Eric Dunham, who's speaking next, might tell you a little bit more about recent studies in primary productivity. So then it also begs the question, what about secondary consumers? And so I'll just give a quick update on something that other people are much more well versed in than me, but I'll give an intro to the next slide. What species are supported by Great Salt Lake microbiolites, or this charismatic megafauna? So if we take a look at a microbiolite hand sample, we can see not only these layers of microbial mat, but we see, you know, little guest stars here. We have this ephedra larva crawling in between the carbonate structure, feeding on the biomass associated with microbiolite. And you also have the brine fly or ephedra casing embedded within this microbiolite structure. So I'm just going to take you through a rough estimation of rough estimation of a food web, wherein ephedra or brine flies are supported by Great Salt Lake microbiolite periphyton. The microbiolites also might contribute to the Great Salt Lake phytoplankton as biomass gets sloughed off of these microbiolites. And so this is just a picture showing some brine shrimp, who I don't know what the genders are of them, but they are living happily next to this microbiolite in a tank that we had in the lab at Montana State University for a couple of years. And so lake phytoplankton can support the Artemia species or the brine shrimp species. And then of course, the brine flies and the brine shrimp support Great Salt Lake bird populations. And the Great Salt Lake supports over 200 species of birds, over 7.5 million birds annually as a part of the Western Hemisphere Shorebird Reserve Network. And so microbiolites represent an important source of primary productivity to the lake. They do not exist in a vacuum. And so going back to the experiment we did with different salinities, we also took a look at Artemia responses to changing salinities. So this plot shows the percentage of shrimp hatched in our little microcosm experiments over a period of about 16 weeks at these 6 different salinities. We can see that at salinities of 8 and 10%, a lot of shrimp were able to hatch. They quickly died off, but there was a steady population that remained. But in salinities of around 15 to 20%, we did finally get some shrimp present in our microcosms after 12 weeks. And then at salinities of 25 to 30%, we never saw any shrimp whatsoever. So that indicates that there is a point, a shifting point at which shrimp no longer are able to survive or are no longer able to feed on microbiolite biomass. So I just want to leave you with some final thoughts here. Yeah, I was very lucky to be involved with a couple of studies in Eric Boyd's lab at Montana State University and our collaborators, of course, Bonnie and Jaimi. But these are some of the final thoughts I have in general on microbiolites. And so microbiolites of Great Salt Lake are a unique and necessary ecosystem. They're a source of primary production, and they represent the largest expanse of microbiolite structures on Earth, on present day Earth. With our lab-based studies, we saw major decreases in primary productivity at salinities greater than 15%, and the species present also shifted, both in the lake, both the south arm and the north arm microbiolites, and in our salinity incubation experiments. Looking at Artemia, they did not even hatch at greater than 20% salinity, but they were happiest at around 8 to 10% salinity. And so the final thought here is that major anthropogenic events such as the construction of a railroad causeway in the middle of a lake cause major impacts to these structures at a microbial level, which can also impact macroecology of the lake. So I have many people to thank. I first like to thank Boyd Geobiology Lab at Montana State University and my PhD advisor, Dr. Eric Boyd. Had a great time during my PhD, and I miss both Great Salt Lake and Yellowstone and the lab very much. Thanks to all of our collaborators, particularly Bonnie and Jaimi at Westminster, and thank you to the funding sources that made this work possible a couple years ago, particularly the Doyle Stevens Scholarship that I received from Friends of Great Salt Lake a couple years ago. So I'm happy to take questions in the question time in about an hour, but if you can think of any others, feel free to email me. I'm currently a postdoctoral researcher at Bigelow Laboratory for Ocean Sciences, so I took a little bit of a turn. I'm currently focused on deep sea subsurface microbiology, but I love thinking about microbiolites still, so happy to answer any questions. And thank you all for having me.
Thank you, Melody. I mean,
Microbialite. I meant thank you, Microbialite.
That's my good name.
That last picture was to die for. I must-
Intense microbiolite observation.
I know.
Definitely wasn't a nap. No.
No. I have a picture of your arm down in a giant core hole too. So, you know.
Yeah, there's a lot of, one of our collaborators, Dr. John Spear at Colorado School of Mines, he has a series of pictures of me and across the world, in Yellowstone, in New Zealand, and in Great Salt Lake with me in odd sampling positions. For some reason, John Spear is always there when I'm in the mud.
I forgot to introduce you to my brine shrimp puppet. And I thought maybe this brine shrimp puppet could introduce Eric. Would that be too inappropriate or not professional?
No, I think that's delightful.
So Eric Dunham, he is a PhD candidate at Montana State University, expecting to graduate this summer. In his thesis work, he investigates how the microbial communities that live under glaciers can survive using hydrogen gas and bedrock materials. At Great Salt Lake, Eric has studied the formation of the mineral dolomite by microbial mats on the lake floor and is currently working on estimating the primary productivity of the lake's photosynthetic cyanobacterial communities. Take it away, Eric.
All right, thanks, Jaimi, or thanks to your brine shrimp friend. I'm just getting to here. Can everybody see this?
Eric down started. Oh yeah, there you go.
Yep.
Okay. So as Melody kind of touched on, I want to talk about a newer project we have going on at the lake that is it's in progress right now, and so I thought what I would do is just kind of walk you through what we are attempting to do with this project, how we do it, and have some. I'm going to end with some sort of wild extrapolation of our existing data, just kind of for fun. So, Melody's talk was really about the microbiolites being the basis for the food webs at Great Salt Lake. And let's see, let's see if my slides will advance here. There we go. So, we know that this entire Great Salt Lake ecosystem is really supported by microbial primary production at its base. We have, these benthic microbiolite communities that feed the brine flies, and we have the planktonic microbes that feed the brine shrimp. Correct me if I got that mixed up, but and the brine flies and the brine shrimp are a critical food source for all the birds that are supported in this. It's a massive wetland habitat, basically. And one of the things that's sort of strange about this is that nobody's ever really estimated, nobody's ever tried to quantify the primary production at Great Salt Lake. And so that's what we're trying to do with this project we have underway now. So primary production at the most basic level is turning CO2 into biomass. I have this chemical equation up here. We have CO2 and water. And with the input of light energy, photosynthetic organisms can turn CO2 and water into sugar and oxygen. Some of that sugar and oxygen they burn to generate energy, just like we do. And then some of that sugar gets shunted into biosynthetic pathways and gets turned into biomass. So that this biomass is generated through photosynthesis is what feeds brine flies, brine shrimp, and those in turn feed the birds of the lake. And so this, photosynthesis is really the foundation of this ecosystem. And so we want to look for a way to quantify the amount of photosynthesis going on in the lake so we can estimate, how much biomass is being generated by the lake each year. So the way we do this is with a microcosm-based approach. And we have these tiny glass vials. They're called serum bottles, and we'll fill them with lake water, which contains microorganisms. And the CO2 that's captured in the headspace of those vials is turned into biomass via photosynthesis. And since all of this is captured in a little bottle, we can hopefully quantify this process after a set duration of time. So I have a photo here of this is me collecting some water to put into our microcosms. Here's Dr. Boyd filling a few of them on our first sampling trip last July. And the way we actually quantify this is we use 14C. So it's a radioactive isotope of carbon. And if we add a known amount of radioactive CO2 to each of these microcosms, that gets incorporated into biomass, just like a normal 12C non-radioactive carbon molecule would, or carbon atom would. And we end up with radioactive biomass. So we can filter out the cyanobacterial biomass from these vials after we've run our experiment. And we can quantify the amount of radioactivity that got turned into biomass. And then we can estimate from there, how much of the total, how much carbon was turned into biomass during the time period under which our experiment was run. So we can use this data, we can use this radioactivity data. We basically look at the ratio of, how much 14C radioactive carbon did we put into these microcosms in the first place, and what percentage of that was incorporated into biomass, and that's how we can get an estimate of how much biomass was generated during our experiments. So the way this works is we first have to go out to the lake. Here's a picture of Libby Foams, who's another member of the Boyd Lab. We go out in the lake, we just collect water and we start filling bottles. And here on the right side, you can see her filling. We have somewhere between 50 and 100 microcosms that we fill on each sample trip. And the first step in this, like I said, is just to fill the bottle with some lake water that contains planktonic cyanobacteria that are fixing carbon in the lake. So after we've filled our microcosms with lake water, next we need to kill half of them basically. And we'll use this to control for any abiotic reactions that may occur in any non-biological ways in which this 14C we're adding could be captured within our filtered microcosm water, giving us a false signal. So what we'll do is we'll boil half of our water samples into jet boil. This should kill any microbial cells in our water samples, and these heat-killed microcosms should be fixing, quote unquote, fixing less CO2 than our biological control, or our biological sample microcosms. So once we've killed half of our microcosms, we next have to find out, how much of this carbon fixation is due to photosynthesis versus other microbial pathways. There are several microbial pathways by which CO2 can be fixed into biomass, and they're not all light dependent. So what we'll do is we'll wrap half of our microcosm bottles in aluminum foil to keep it dark inside, so that as we incubate these during our experiments, we can get a difference between the amount of CO2 fixed in our light exposed microcosms versus our dark microcosms. And we'll be able to use that difference to estimate how much carbon fixation is due to photosynthesis versus other potential pathways. And finally, the last thing we want to sort of discern with this project is how much primary productivity is being carried out by planktonic microbes, which are those that are, these are floating in the water column, versus the microbes that are making up these mats and microbiolites on the bottom of the lake. And so the way we do this is we set up, a set of microcosm experiments where we just use lake water. And then we have another set where we use filtered lake water. So we filter out any planktonic microorganisms, and we add samples of this microbial mat from the bottom of the lake. And so these microcosms will give us a sense of, how much photosynthesis is being carried out by the mats on the bottom of the lake, independent of all the bacteria that may be floating in the water column. So as you can see here on this diagram I've been putting up, this kind of escalates in complexity pretty quickly once you start adding experimental conditions. And so we end up having, about 48 microcosms, I think, per sample site. So the last step in this is we need to spike them all with 14C labeled CO2. We can't do that out in the field or right next to the lake, which would be convenient. So we take all these microcosms on ice back to our lab, and we spike them with a a known quantity of radioactive CO2. And once we do that, we incubate them for 30 minutes out in the sun, and we use a water bath that we calibrate to the temperature of the lake water that we measured on sampling day. This usually happens, either, well, it's been the day after we take our samples typically. So we drive down to the lake, we'll collect all of our samples, we'll drive back to Bozeman, we'll get back home around 10 to midnight, wake up the next day, go to the lab and add our 14C, and we'll set out these water baths for half an hour in the sun to let these microbes, do their thing and fix as much carbon as they're going to. We'll then flash freeze them on dry ice or in a minus 80-degree freezer to kill all the cells, halt all biological activity, and then we can process them sort of at our leisure. So the way we do that is we add acid to drive off any CO2 that's still dissolved in the water. Then we'll filter out the cells and we'll wash them a few times with pure water in the lab to wash out any dissolved, if they make waste products that this 14C has gotten into, we'll wash those off. And then what remains on our filter is biomass. And we assume that whatever radioactivity is associated with that filter is radioactive carbon that has been assimilated into biomass during the course of the experiment. So the things we can address with this project design are, using our live versus heat killed controls, we can find out what's a biological source of carbon fixation in the lake versus an abiotic sort of just chemical reaction that might result in CO2 precipitating. We can determine what's a photosynthetic carbon fixation reaction versus a light independent microbial metabolism. And we can estimate the primary production of both the planktonic or water column microbes and the benthic or lake floor microbiolite microbes. And so I mentioned this a minute ago, but we have these sort of branching, with each variable we want to test, we double the number of microcosms we test, and we typically like to have 3 replicates for each experimental condition so we can get a nice average and an idea of the, the sort of the spread in our data. So this ends up being 24 microcosms per sample site. We sample 2 sites per trip, and we actually have, we do a couple other experiments while we're out here with different carbon sources. So this ends up being a lot of bottles each sampling trip. So here's how it kind of works on a geographic level. We go to Great Salt Lake. Our sampling site is here on Antelope Island. And we have 2 sites, like I mentioned. One is up on the north of the island here. This site has a lot of these microbiolites, sort of like Melody has shown. I think this was actually a sample site she used in her experiments a couple years ago. And so at this site, we sample the planktonic or water column microbes and the microbiolites on the lake floor. Our second site is out here at White Rock Bay. And this site is a lot more, it's flat, it's muddy, and there are no microbiolites here. So we use this as a second sampling site for just the planktonic or water column community. So after we go here, we sample, we go back to the lab in Bozeman and we add our 14C, and then it takes a few days to process all these samples. What does the data look like? So what I'm going to show you here on this graph, on the y-axis, we have femtomoles of carbon assimilated per milliliter per hour for this planktonic community. So a femtomole is a tiny, tiny amount, but we're dealing with these little tiny bottles filled with lake water, and they're only incubating for half an hour. So we're just getting a very small snapshot of the amount of carbon that can be assimilated by a small portion of the lake in a small amount of time. And you're going to see blue bars and orange bars here. The blue bars represent the amount of carbon we see assimilated to biomass in our dark reactions, and the orange bars represent the amount assimilated in our light reactions. So you can see here, this is about what we might expect to see for a photosynthetically dominated community. We can see that these planktonic cyanobacteria are light dependent, they're photosynthesizing. and we see a lot more carbon fixed when we expose them to light than when we don't. There's some variations between our two sites, and we can potentially investigate that as our experiment goes on. But really, this basically lines up with what we want to see. We want to see photosynthesis happening, and this is good evidence of it. When we do our microbial mat experiments, one thing I want you to notice right away is that the scale is different on our y-axis. Now we're dealing with picomoles. This is still a tiny amount of carbon, but it's 1,000 times as much carbon as a femtomole. And so what you're going to see, even just looking at this axis without seeing the data, you can see that the microbial mats are on the order of 1,000 times more productive than the water column. And so what you're going to see here, we normalize, this is picomoles carbon per gram dry weight of microbial mat material. And you can see here again that this is a photosynthetic community. There's a lot more carbon assimilated when it's exposed to light, which is, again, what we would expect. So I've been showing you a lot of, this is femtomoles and picomoles. This is per gram dry weight per hour. These are kind of abstract numbers. And so I wanted to spend the last couple of slides just trying to put this in context for people. And so I'll warn you ahead of time, these are kind of, it's borderline irresponsible to project this far based on how much data we've collected so far. We want to be able to, eventually with this project, we want to create the first estimates of primary productivity on an annual basis for the lake. That's obviously a really complex calculation. And what I've done on these next couple slides is done a really back of the envelope simplified version of the calculations we'll be doing, just to try and give us a ballpark figure for how much carbon is fixed by these photosynthetic communities. That carbon becomes biomass that can be fed up the food web of Great Salt Lake, feeding all of our brine shrimp, brine flies, and the birds. And so I'll dive into it here. As far as the planktonic side of it goes, from our data, we have averages between our two sites of about 160 femtomoles per hour in the dark and almost 1,000 femtomoles carbon per mil per hour in the light. So per a paper from 2005, we get an estimate of the total volume of the south arm of the lake. So all of these calculations, I'm just working with south arm figures since we have not done these experiments in the north arm. And you can see just by the, by the pictures Melody showed, the north arm is a dramatically different environment, and I wouldn't expect the measurements we're getting in this experiment to be applicable to the north arm. So the volume of the south arm is something on the order of 1 × 10⁻16 milliliters. So that's a huge number. I'm sure you can guess. It's sort of, again, hard to wrap your head around. But we'll just bear with me for these calculations, and we'll try to make some sense of it eventually. We can get a very rough estimate for the hours of daylight per year at Great Salt Lake, just based on its latitude and climate. And so we get about 4,300 hours of daylight per year and 4,300, 4,400 hours of darkness per year. And so I'm going to use these hours of daylight with our daylight productivity estimates, hours of darkness with our dark productivity estimates, and make some wildly borderline irresponsible calculations. And we get to carbon fixation rates of about 56 megamoles of carbon per year. So again, this is just a huge number. We're going to have to wait 1 second to make some sense of it, because I want to go into the benthic mat productivity figures here too. So again, we're making some wild extrapolations. We have our average dark productivity now is 400 picomoles of carbon per mil, or not per mil, sorry, that's per gram dry weight sediment per hour. Light productivity is 2,000 picomoles per gram per hour. A 2014 paper estimates that benthic mats cover approximately 700 square kilometers of the south arm. We have a 2011 publication estimating this is kind of, again, these are wild estimates, but we're looking at chlorophyll levels of approximately 900 milligrams per square meter. And the way we can use these numbers is that our measurements in this experiment, we can correlate the amount of chlorophyll to the amount of dry weight of mat material. And so again, to make a wild extrapolation, we get our measured rates should correlate with annual carbon fixation of about 39 megamoles of carbon per year. Okay, so we have these big numbers. What do they look like? What can we, how can we relate to this? What do we know about the primary productivity in the lake? So our planktonic productivity estimate was 56.7 megamoles per year. Benthic mat productivity was 39 megamoles carbon per year. And the sum of these numbers gives us 95.7 megamoles of carbon per year. How much is 95.7 megamoles? I'm sure you've all been asking this since I first started talking about this. So 95.7 megamoles is about 4,200 metric tons of CO2. Again, this is hard to wrap your head around. 4,200 metric tons is the mass of about 40 blue whales or 700 elephants. 4,200 metric tons of CO2 is about the amount of CO2 released by a car after it's driven 10.4 million miles. Put another way, it's about the amount of carbon fixed or taken up by 70,000 tree seedlings as they grow over a period of 10 years. So this is, it's a massive amount of carbon being, turned into biomass, taken out of, this is a greenhouse gas being taken out of the atmosphere, turned into biomass in the lake every year. And so, I hope that these numbers are sort of something to help you get your head wrapped around this. And what I want to do for this project in the future is, I mentioned we want to get more accurate estimates here. We think these will actually turn out to be low. So all of these calculations are based on data from our July sampling trip. I actually haven't finished processing all of our microcosms from our October or January trips. And we're actually doing a final trip in April. So the idea here is to get productivity estimates for the summer, the fall, the winter, and the spring. And once we have all that data, we will, refine our calculations we've been doing and try to get a more accurate estimate of this productivity changes throughout the year, and we want to get an estimate of how much productivity is occurring throughout the year in Great Salt Lake. One thing I can tell you that we know is that microbial biomass in the lake tends to be higher in the winter due to the absence of the brine shrimp and the brine flies, which are the primary predators of these cyanobacteria. So that's why I think these estimates I've gotten right now will turn out to be low. I can tell you from our January sampling trip just a few weeks ago, the first indication that we have a lot more microbial biomass in the water in January is that the filters we used to filter the water got clogged up way faster. In July, I was able to get something like 500 mils to a liter through a filter before it just became too hard to filter any more water. And in the winter, I was only able to get between 150 and 200 mils through a filter before it was totally clogged. So yeah, I'm really excited to continue this project. Like I say, the estimates I gave you here are, in scientific terms, these are wildly irresponsible predictions to make because we really haven't constrained all the variables that go into these calculations very well. And so by the time we get publication quality data, I hope that there will be a lot more work going into this, and I hope we'll have a lot more accurate estimate. And ideally, it's going to be a lot higher than what we've seen today here. So with that, I want to thank especially Dr. Boyd, who's my advisor in graduate school. Libby Phones is another member of the lab, actually my girlfriend, who came out on these sampling trips with me. And from the Great Salt Lake Institute, I want to thank Jaimi and Bonnie and all of the student assistants who come out on these fieldwork expeditions and make this work possible. And with that, I will turn it back over. Let's see if I can figure out how to stop sharing my screen, and I'll turn it back over to Jaimi.
Thank you, Eric. Awesome. Amazing. Really cool to see some of this new research coming out. Thank you so much.
Yeah, thanks for having me here to present it.
Now, usually I don't like to share my screen in the middle. I want to hear other people talk, but I have so many things to say about brine shrimp that I'm going to share my screen, and I'm going to talk about brine shrimp for just a minute until we invite Phil Brown to join us. So when we were editing this book, it was a little bit hard to get really excited about particular sentences that people had written. We had read 16 chapters of sentences, and this one really struck me. Extraordinary is an insufficient term to describe Artemia franciscana, an arthropod well adapted to extreme conditions where they serve pivotal roles in the Great Salt Lake ecosystem. The reach of their influence extends well beyond the boundaries of Great Salt Lake. We all have a lot of fun catching these in little cups when we are out at the lake. This was a summer camp that we were part of, and sometimes it's hard to describe, it's hard to explain the brine shrimp that are coupled up in these cups to second graders. This might be, you know, something kind of a typical life cycle that you would see of brine shrimp, where these incredible dormant cysts hatch in the springtime in the really great conditions, and then they go through lots of different instars, or they molt lots of different times until eventually they become adult males and females. You can tell the males from females because they have these claspers that they use to grab onto the females. If you look at them under a microscope, I think we already told you they do have two penises. And then the female brine shrimp, again, super incredible. They can either release live young if the environment is good, or they can release dormant cysts that have a hard outside shell on them. I've heard somebody describe these cysts kind of like an M&M, that they have a soft embryonic center and a hard outside shell. And actually, this life cycle, I'm so glad that in the chapter on brine shrimp in our book that Brad Martin, Phil Brown, and Thomas Bastiles, who work at the Great Salt Lake Brine Shrimp Cooperative, they edited this life cycle to show just how amazing these dormant cysts are. So when these dormant cysts are released by the females in the fall or early winter time, they're actually in it's called diapause. It's a suspended state of metabolism. And so if you took those cysts and you put them in exactly the right salinity water and the right food and the right light, they wouldn't hatch. It's one of their cool adaptations, so that in those Indian like in those late winter days that sometimes get really warm at the lake, they don't hatch and then die when it freezes again. This is really important for the brine shrimp industry. They're taking those dormant cysts and they're disinfecting them and drying them and sending them around the world for use in commercial aquaculture. I think they look really cool under a high-powered microscope. This is what they would look like. They're, you know, very tiny, 250 microns, and they just look kind of scary. This is a very young shrimp. You can see them kind of get bigger, and they start to grow thoracopods that they use to filter algae out of the water. I know that this is a teenager brine shrimp. You can tell because down by the tail it says FU. This is a juvenile brine shrimp. I think this picture, this is a adult male brine shrimp with his big claspers that I think makes a really wonderful portrait. Brine shrimp do dance. They make very good food for fish because they do have that movement. Again, very tiny, tiny, tiny. Each one of those little dots that looks like a grain of sand is a brine shrimp cyst. You might have seen them on the back of comic books as sea monkeys. Sea monkeys were actually a different species, but we're pretty similar. We don't really understand where the crowns and the nuclear family came from because that's not really how our brine shrimp are, but it's kind of fun to think about. It's fun to dress up like brine shrimp if you need a great costume to wear to this next Halloween. Maybe we can all be together. And they're so cool that they've even been on South Park before. I highly encourage you to go check out this episode of South Park. And you might wonder, I didn't know about the brine shrimp industry when I started working at the lake. I started working with the brine shrimp industry in 1999 as a brine shrimp biologist. And they actually have all of these planes that are surveying 200 or 300 feet off of the surface of Great Salt Lake, and they're looking for our floating cysts of brine shrimp. They look like an oil streak, and we call them streaks or slicks, just like oil. You can see this is a large boat that's in the center, and all of this brown material are those brine shrimp cysts that the harvesters are looking to capture. Those planes call down to high-speed boats, and they race across the surface of Great Salt Lake, where they can then claim their territory. They carry these permits on their boats, and with each of those permits, the harvester has 300-yard radius around the boats that they can harvest. And then they deploy, this is oil containment boom, exactly the same stuff as you would capture an oil spill. It's about a 3-foot floating plastic sheet that hangs in the water, and they can use those to encircle and gather up those cysts. When they do that, the raw cysts that come off the lake, they're wet, and they kind of feel like wet sand in your hand, and they don't often smell very good either. And then they pump those cysts on board their boats in these big filter bags. Each of these filter bags can hold up to 2,000 pounds of cysts. So you can imagine here, 1, 2, 3, 4, 5, 6, 7. I mean, these are like pretty incredible boats that are out on the lake harvesting these shrimp. Some people wonder what it's like riding in one of those. I feel very lucky that I was invited onto some of these boats. And here, Brad, I think, is on this call. I think I saw him. Brad is cooking some dinner, and my friend Jim Hopkins, who knows, he's eating dinner, and you can see the lake behind us, and there's bunks in the boats. Sometimes they'll even stay out on the lake. And there's a big barge too. It's like a city on this lake with all of these brine shrimp boats that are providing infrastructure for the industry. And then obviously, our fishermen need to eat, and they need to be able to live, and they have resources on these boats. I feel super lucky that I got to learn from all of these characters. This guy, Dave, he used to wash all of the salt off my windows. He would yell at me because I never washed my windows, and they get very salty. Mark Jensen, he is one of these legends of the brine shrimp industry, here doing a back scratcher. I think this was a 1972 cover of Powder magazine, but in the winter he would come to Great Salt Lake. He's a local Utahn, and he would go to Great Salt Lake and harvest brine shrimp. And he actually taught me a whole bunch about shrimp out on Antelope Island, from a perspective of brine shrimpers that sometimes differs from other people. This is my friend Pat Leemacher water skiing on Great Salt Lake. I don't recommend water skiing on Great Salt Lake, but brine shrimp harvesters sometimes have a different take. He was a mechanic, and I mean, I remember him falling in the water and just, mucus like just pouring out of his nose after he got all of that salty water. Crazy fellows. And I think that I've taken all of this time to talk about this because I think there's a really lovely story about cooperation. We didn't just, these fishermen are in a flotilla, they would hook their boats up, and when there was nothing going on, and would, have like little parties out there or whatever. But I think that the brine shrimp industry and the state of Utah is a really great and shining example of how an industry and management can work together with a common goal of having sustainable brine shrimp harvest, not just for shrimpers that make money from that and that it's used in aquaculture, but also for our birds. And the brine shrimp harvesters, they actually came to the Utah Division of Wildlife Resources and said, hey, we're looking at this graph right now, right? And this was kind of early on in this harvest. And you can see that there's in this harvest industry, and you can see there's lots of ups and downs and how many raw pounds of brine shrimp is harvested off of Great Salt Lake. And the industry was really worried about that, particular, some of these very low years, that's very hard on an industry. And so they came to the Utah Division of Wildlife Resources and said, hey, we think that we need to be managed in order to have a sustainable harvest. And the Utah Division of Wildlife Resources responded and eventually created the Great Salt Lake Ecosystem Program that manages this harvest for brine shrimp and people. And I think that the state of Utah has done, and I don't say this very often about the state of Utah, but I think they've done a really beautiful job of managing this harvest adaptively. So they, the harvesters can harvest brine shrimp out of Great Salt Lake only until a certain level. When the number of brine shrimp cysts reach 21 cysts per liter, they have to cut off harvest. And I want to pay particular attention to this amazing graph. I hope you can see it really good. In these last few years, it actually appears like because of this management scheme and the industry working with the DWR, that there's a more consistent and even optimized harvest. And that always helps, again, our economy and our birds. So I think that our state crustacean should be brine shrimp. I think that instead of polar bears on a Coke can, that we should have brine shrimp on our Diet Coke cans, because we are in Utah, we all love Diet Coke. And I have made formal calls for our mascot to change at Westminster. Nobody's responded yet, but I have hope that it will be the state crustacean, and that we will change our mascot. Can you tell I love brine shrimp? I love brine shrimp. And so right now, I would like to bring out Phil Brown. Oh, here, Phil. I'm going to introduce you with the brine shrimp, my puppet. Phil Brown has a background in limnology. He graduated with a BS in watershed sciences from Utah State University, where he had the opportunity to work with disparate ecosystems of the Sawtooth Mountains and Great Salt Lake, and gained an appreciation for the diversity in aquatic systems. I can't do both. I'm sorry, Phil. I was going to introduce you all. I can't do both. He graduated from Oregon State University with an MS in fisheries science, studying the effects of agricultural water withdrawals on river invertebrates before making the jump back to hypersaline. I love that you jumped to hypersaline. That's awesome. He has been involved in the Great Salt Lake ecosystem and Artemia population monitoring since 2008. First as a biologist for the Division of Wildlife Resources, and currently with the Great Salt Lake Brine Shrimp Cooperative, where he is involved in the long-term monitoring of Artemia population, nutrients, phytoplankton, and the lake's deep brine layer. He has dragged his young twins to the shores of Sawtooth Mountain Lakes and Great Salt Lake, where they remarked that this one's too cold and this one burns my feet, and thereby seem to be gaining their own appreciation for the diversity of aquatic ecosystems. So before you start though, our kids are the same age, right? And I've been saving this joke. Hey, Phil.
Yes.
Knock, knock. Oh, sorry. Knock, knock.
Who's there?
Brine shrimp.
Brine shrimp poo.
Ha ha ha. You said brine shrimp poo. My kid.
Yeah, it's probably Probably a good thing my kids aren't in the room, or I'd be hearing that to
I'm
no end.
stealing that. I'm totally going to steal it too.
That was from my you got to attribute it to my 8-year-old daughter, Cora. Thanks, Phil, for being here. And I mean, I want to say again how much I appreciate the Brine Shrimp Co-op participating in this book. It was something that we really felt was important to have an industry perspective, where a lot of times Brine Shrimp comes from, these perspectives come from academia. So thank you for coming. We're really stoked to hear more about Brine Shrimp.
Hey, thanks. Let me start my screen share.
It looks great.
Okay. There we go. Okay, well, thanks, Jaimi. This presentation, we're going to try to touch on most of the topics that we address in that chapter of the book, and it's going to try to cover a lot of ground and, a minor amount of detail, but more detail of these topics can be found in that book chapter. So, as you mentioned, I'm from the Great Salt Lake Brine Shrimp Cooperative. We are a cooperative of 17 individual companies which harvest the overwintering eggs known as cysts that the brine shrimp leave behind in the lake each fall. And cysts are used in aquaculture worldwide. This is a sustainable fishery regulated by the state of Utah using an adaptive management program. And so I'm an aquatic biologist with the research and development arm of our company. And the status of the brine shrimp population and the health of the lake is crucial to us. And we maintain a year-round monitoring program that samples the lake in multiple locations. And we examine the brine shrimp population structure. We measure variables such as nutrient levels and the abundance and type of algae upon which the brine shrimp feed, and the characteristics of the cysts themselves. And we have data going back to the late 90s and have been doing this intensive year-round surveying for the past decade. And on the side topic, we're really grateful that you showed that video of the giant brine shrimp attacking Salt Lake, because it's a common misconception that that was a spoof film. It's actually a documentary. And the monster brine shrimp hatches from a single giant cyst, and every year our brave fishermen go to great lengths to try to capture that giant cyst and get it off the lake before this disaster can happen again. See?
That's
It was a
brilliant.
close call this year. That one was about ready to hatch. So, we don't want to brag too much about it, but maybe when you get the chance, thank your local brine shrimp fishermen. Okay. Brine shrimp is the common name for the genus Artemia, and as we know, they're not true shrimp. They are crustaceans, and they reside on a narrow branch of a phylogenetic tree that contains Daphnia and fairy shrimp, which look a lot like Artemia, but tend to live in isolated freshwater pools and ponds. Artemia are small. They only reach about 10 to 12 millimeters in length, depending on the food supply at the time, and they're free swimming within the water column. And they're planktonic, meaning that while they are able to move short distances under their own power, they can't overcome currents and must simply go where they're taken. They eat single-celled algae that float about in the water column, known as phytoplankton, by filtering it out of the water as they swim. And they're very good at this. At peak population densities, they can filter the entire volume of the lake in less than a week. Artemia can handle a wide range of salinity. Physiologically, they can tolerate salinities below 1% and above 20%, though they tend to be healthiest at the low end of the salinity spectrum. However, other organisms are able to occupy that low range of the salinity spectrum as well, and this creates a problem for Artemia because they're not good at avoiding predators and they're not good at competing for resources. So as a result, Artemia kind of need to have the place to themselves. Exclusion of predators in this lake doesn't occur until about 9% salinity, when the predatory insect Trichocoryxa, or coryxids as they're commonly called, can no longer tolerate the salinity. And the Artemia also require a phytoplankton population that isn't dominated by inedible forms of phytoplankton such as filamentous cyanobacteria and large varieties of diatoms, and that has been observed to occur at salinities below 11% on the lake. Artemia can also become stressed in extreme salinity. Above about 18%, we've seen declines in the population in our data. So the optimal salinity range for this population is therefore between 11 and about 18%. And as a result, Artemia tend to thrive in Gilbert Bay, which maintains this general salinity range most years. Gunnison Bay, north of the railroad causeway, is far too salty for a healthy population in all but the rarest of years, and parts of Farmington Bay can maintain a small Artemia population during years when the water level is correct for that. So I should provide a description of what an Artemia cyst is for those who aren't quite familiar, since this stage of the life cycle is key for their ability to live in Great Salt Lake. When we say cysts, we're talking about an Artemia embryo. In the gastrolith stage of development, which means the cells have begun to organize into layers that will later differentiate into specific tissue types. At this stage, it's composed of about 3,000 individual cells, and they're protected by a durable, semi-permeable, semi-flexible shell with multiple layers. The outer layer, you can see here, is pigmented, and it shields the embryo from UV damage, and it provides flotation for the cyst. The inner layer, the embryonic cuticle, is dense, it's rich in chitin, and it's impermeable to most substances, though water, carbon dioxide, and oxygen can pass through. Artemia produce cysts in response to unfavorable conditions such as starvation, cold temperatures, rising salinity, cues that their habitat is entering a phase which can be lethal to the adults. Cysts are produced essentially to endure difficult times. Very quickly, upon release into the water, the embryos enter a state of dormancy until conditions become favorable again, in which case the cysts will hatch out as the youngest free-swimming phase, known as nauplii. When conditions are favorable for the adults, the Artemia adults can bypass cyst production altogether and release the nauplii directly as live young. I wanted to provide a brief look at the yearly population cycle of Artemia in Great Salt Lake from our long-term data set. And this year in particular is representative of the general patterns that we see repeated year after year in the system. So the red line we see on the graph is the Artemia population size given as the number of adult Artemia measured per liter of lake water. The green line is the phytoplankton population size measured as the density of photosynthetic chlorophyll A pigments in the water. The light shaded gray is the production of live nauplii. The dark shaded gray is the production of cysts by the population. So in January and February, cysts are the only Artemia life stage in the lake because winter is too cold for the adults and they perished back in December. Hatching begins in March and April as the temperatures warm and river inflows tend to freshen the upper water layers of the lake. And the Artemia have an abundant food supply that has grown over the winter. The Artemia grow rapidly, produce more nauplii, and the population peaks sometime in May or June. And so for a brief time, it's really good to be a brine shrimp. However, the conditions turn quickly. The population rapidly exhausts the food supply, consuming it at a rate much more quickly than the phytoplankton can subdivide and reproduce. And so by the beginning of June, there's hardly any phytoplankton left in the lake, and the population begins to starve and produce cysts in response. Only about 10 to 20% of the adults survive that starvation period, and the phytoplankton, which are now freed from so much grazing pressure, can begin to grow back. So for the rest of the summer, the dynamic is much more stable. The Artemia are hedging their bets here by producing a mixture of nauplii and cysts, and their population doesn't expand to anywhere near the level seen towards the beginning of the year. And so in September and October, as winter approaches, more resources are diverted into the production of cysts, and the cysts remain in the lake and washed up on the shoreline throughout winter and begin the life cycle again in spring. Now, Artemia are important to both wildlife and people because of the ecosystem services that they can provide. One of these is control of eutrophication, which is the enriched state of a water body that results in high productivity. Great Salt Lake is at the terminal end of a very large watershed, so it naturally has a high nutrient content and receives additional nutrients from human activities in the watershed, and so it's very productive. In winter and spring, when the Artemia are absent, the phytoplankton uses these nutrients to grow at very high density. However, when the Artemia population peaks, they can consume that phytoplankton within a few weeks and convert that algal biomass into Artemia biomass. Water clarity goes from as little as 8 inches to more than 20 feet. This peak Artemia population each year is really brief because they quickly die back from starvation once they've consumed all of the food. And so during this brief peak, up to 8 adult shrimp per liter of water have been measured, once by state biologists and once by our own program. When this occurs, there's an incredible amount of Artemia biomass in the lake. How much? Well, if you take that 8 Artemia per liter and multiply it by the number of liters within Gilbert Bay, and then multiply that by the approximate weight of each brine shrimp, you get a mass that's equivalent to that of about 1.4 million people, assuming each of those people weighs 150 pounds. And that's essentially the entire population of the Salt Lake Valley. Now, remember that the Artemia population stabilizes at a more lower sustainable number after that first starvation period. And if you use that lower, more stable number instead, you still derive an estimate of about 210,000 people, which is just Salt Lake City proper at that point. So, Eric, you're not the only one who wanted to extrapolate to get a big number. And we thought this was interesting. And it's probably something to remember if you ever hear anybody say that Great Salt Lake is just a dead, lifeless waste. It's not. And this concept's a bit more enjoyable to see than to hear. So we have some camera footage from a camera lowered to the bottom of the lake that we were trying something out for a different research project. And I think it shows the Artemia population rather well. So let me start this. This is at the deepest location of the lake that we sample. And it's during a time period where the Artemia population is high, but it's not at its peak density for the year. And so we're lowering this camera down through about 6 meters of water until it hits the deep brine layer at the bottom of the lake and continues for another meter, meter and a half. There's that salinity distortion entering the deep brine layer, which is anoxic, full of hydrogen sulfide, decomposition. And now fast forward to when we go back up through that deep brine layer and return to the surface. There we go, back into the oxygenated water layer of the lake. And something to keep in mind is that you're only seeing shrimp that are a few feet away, what the camera can see. Keep in mind that Gilbert Bay is 35 miles long and about 15 miles wide. So if you extrapolate what you were seeing here across a water body of that size, it becomes apparent that there's a lot of life in that lake. And up to the surface, and there's Brad Martin. Hi, Brad. Okay. So, as we can see from that, the Artemia population really is a vast nutritional resource for organisms that are capable of tapping into it. No fish or aquatic insects are present at these salinities, but there are several avian species that do feed on Artemia, and the most notable of these is the earred grebe, which uses the open waters of Gilbert Bay as a staging location during migration, particularly during fall and winter, when there can be up to 4.5 million of them on the lake recorded by avian biologists. They feed exclusively on Artemia and are able to dive deep below the surface to find them. And flotillas of grebes, as you can see in this picture, can literally stretch for miles across Gilbert Bay when they're doing this. Other waterfowl, such as green-winged teal and northern shovelers, will dabble at cyst streaks in the water when freshwater wetlands are frozen over. And California gulls will even peck at Artemia accumulations at the surface. And while Artemia are not the primary food source for these birds, they can provide supplemental energy during difficult periods. And humans are another organism that can benefit from Artemia as a food source. Artemia nauplii are a crucial feed for the critical early stages of a number of aquaculture species, especially several varieties of table shrimp and prawns. Artificial feeds can't substitute for Artemia here. The young prawn larvae require the swimming motion of the Artemia to trigger those predatory feeding instincts, The nauplii are the correct size for those life stages to consume. They're highly digestible, and they can be used to deliver nutritional supplements to the larvae as well. So aquaculture is becoming increasingly important across the globe as global demand outpaces wild harvest. And aquaculture is currently on pace to overtake the amount of wild-caught seafood on a global scale. So this situation's not likely to change. Okay. So Great Salt Lake can be a hostile environment towards any organism that's attempting to live there. Some organisms like migratory waterfowl can escape, but Artemia have to dig in and deal with it year-round. And they face lethally cold winters, lethally hot summers, particularly on the shoreline where many of the cysts wash up. And no matter the season or the habitat, there's always salinity to deal with. And few species in the animal kingdom are adapted to this kind of extreme. So how do Artemia thrive here? They do it through a diverse series of adaptations which mitigate environmental stresses through their physiology and avoid environmental stresses through life history strategies. Free-swimming Artemia, which are the nauplii, the juvenile, and the adults, face high salinities, low oxygen levels, and very warm water temperatures. They survive the salinity by actively pumping salt from their bodily fluids to maintain a relatively fresh internal salinity. It takes a lot of energy to pump salt against an osmotic gradient. And so this process is energetically expensive for the animals. And the higher salinity, the more energy it takes to maintain their internal salinity. Hypersaline water also holds a lot less oxygen than fresh water, and Artemia are capable of quickly increasing their hemoglobin supply and making types of hemoglobin that are more effective at binding oxygen. Artemia's adaptation to winter temperatures is to produce cysts, which enter a dormant state where metabolism and cellular processes are almost entirely halted. They ride out the cold winter until spring, when internal controls and environmental conditions prompt them to hatch. This sequence of dormancy is known as diapause and quiescence, and it's a very complicated realm of cellular biology that was figured out from about 50 years of detailed research from some very good scientists. So cysts have to cope with more than the low winter water temperatures. As they're released into the water, the high salinity pulls water out of them, dehydrates them. Cysts are washed onto the shoreline, and those that are dehydrated even further by summer heat. And the dehydration itself would kill most animal species regardless of their life stage. But the cysts, particularly those on the shore, also have to contend with freezing air temperatures, extreme summer heat, and constant UV exposure. So first of all, the Artemia shell blocks UV light. Second, they have an unusual set of sugars inside the embryonic cells, such as trihalose, that protect the embryo from dehydration damage. Dehydration causes cell membranes to rupture because the water molecules act as a structural component of those membranes, so when it's removed, it damages them. But in Artemia, as the water leaves, those sugars take their place and hold those membranes intact. And at that point, the dehydration itself becomes protective for those cysts, because if they contained a lot of water, the freezing of that water would lead to ice crystals that would damage the membranes. So being dehydrated works to their advantage. And third, there's a category of organic molecules known as heat shock proteins that protect membranes and other proteins within the cell. Proteins perform a lot of critical cellular functions, but they only work properly when they are maintained in that specific three-dimensional folded shape. And so dehydration and heat will distort the shape of those proteins and render them useless. They won't function properly anymore. Heat shock proteins act as what they call molecular chaperones, and they protect that protein structure, so when the cysts are rehydrated, everything works properly again. So these extremophile adaptations are why Artemia is so well suited for aquaculture and why they're harvested. Because the cysts are durable and in that dormant state, they can be harvested, cleaned, dehydrated, packaged, and shipped across the globe for aquaculture use. They have a very long shelf life, and they hatch very easily for aquaculture operators when they need them. Therefore, the lake's productivity essentially has a global impact, bringing energy and nutrients to aquaculture farms across the globe, which then utilize that to bring energy and nutrients to a large number of people as a food source. So the importance of the Artemia, given this sustainable management of the resource, is critical, and it was great to hear Jaimi talk about this in the introduction, because it is a point of pride for us, for those of us involved on the lake, and it's required a unique partnership between different interest groups and organizations. So the harvest industry expanded very rapidly in the 1980s and early 1990s, and that led to concerns of overharvest. And so in the early 90s, the industry worked with the Utah Division of Wildlife to increase the scientific knowledge of the Artemia population on the lake and try to begin a science-driven management approach. And the resulting technical advisory group included scientists from the industry, from DWR, and from university researchers. A management plan was developed that set that optimum 21 cysts per liter to be left in the lake so that there would be a strong Artemia population to start again next spring. And one of the successes of this is that the management plan is adaptive, meaning that it is reassessed every year as more data is obtained. And as a result of this collaboration, this lake now has a 25-year record of scientific data collection, sustainable harvest, and functional working groups between these different stakeholders. So part of that sustainable management requires increasing our understanding of the Gilbert Bay aquatic system in sometimes in unexpected ways. For instance, we collect a lot of data in our research and monitoring program, and one example that we've done recently is the use of DNA to describe the phytoplankton population. This is something we began several years ago, and we found some very interesting results from it that I won't go into in any detail for the sake of time, but we found hidden diversity within the phytoplankton community. We found assemblage patterns that correlate with the location on the lake and different lake conditions. And we found previously unknown seasonal patterns in individual taxa. And additionally, tying in with Melody's microbiolite work, about 2.5 years ago, during a small sampling, we used these DNA methods to detect microbiolite chloroplast DNA in the water column and within the Artemia near the microbiolite fields. And so that demonstrates that algal cells from the microbiolites do enter the water column and are consumed by the Artemia. And for us, this was a useful reminder that, as with other lakes, the open water and the benthic systems of Great Salt Lake are energetically connected. And so finally, despite this wealth of research, there are a number of issues that Great Salt Lake faces going into the future. As a terminal lake, it's under distinct pressures. There is substantial agriculture and urban areas within its watershed, and that influences the quantity and the quality of water reaching the lake. Great Salt Lake is also exposed to global forces of change with higher temperatures, ultra precipitation forecasted for the future, and some of which is already occurring. These threats are real, but we have reason to believe that they can be mitigated and perhaps some of them even avoided. And there are two main reasons for this. The first is that we already have that cooperative framework already in place, which includes a lot of scientific expertise, motivated stakeholders, and a growing appreciation for the importance of Great Salt Lake. Second, this cooperative approach has already addressed some prior concerns on the lake, including sustainable management of the harvest, progress towards water quality standards for the lake, and even a management plan for the recent railroad causeway bridge that was constructed on the causeway across the lake, which provides a rare opportunity to try to mitigate salt balances across the lake as water supply comes into question. So it's a continual effort by those who care about the lake. And there are a substantial number of people who do, as events like this webinar show. So if we're successful, and there are reasons to be optimistic that we will be, Great Salt Lake will continue to be an important functional ecosystem going into the future. Thank you.
Thank you, Phil. Pretty extraordinary little creatures. I don't even know where to start. I want to talk about brine shrimp all day, every day. But let's start to, I guess, I do want to address, there are lots of macroinvertebrates, a wide diversity of macroinvertebrates that we find in and around Great Salt Lake that we haven't talked about today. I wish we could talk about all of those. Dave Richards was talking, commenting about, you know, the midge larva, fly larva, and how much biomass is really in Great Salt Lake. And we just didn't have enough time to talk today, but thank you, Dave, for putting all of those questions and comments in the Q&A. Geez, Bonnie, do you want to, I don't know where to start, help.
On the questions.
Yeah.
Eric and Mel and I have been answering some of the questions. I don't know, can everybody see the questions or do we need to read them out loud? Can only the panelists see the questions?
I think that they can. I think they can see the question.
So under answered questions, we were getting some really good methods questions from Wayne Wurtzbach, who's worked in this system for a long time, and from Aaron Oren, who Aaron has worked on the Dead Sea and other salty systems around the world. And he's joining us from Israel, which is wonderful. It's like, I don't know, it may be 4:30 in the morning there now. What time is it here? 4:45. So he's up early to talk to us, and he's asking us some really great questions about methods. And so we've been trying to answer some of those specific questions. One of the good questions was about the age of the microbiolites. And I've had lots of discussions with the folks, particularly Mike Vandenberg and Elliot Janecki, who work at Utah Geologic Survey. Wayne was asking about the age of these structures, and Jack Oviatt has looked at the ones at Lakeside, as have other people that were pre-Lake Bonneville. We know the modern ones are likely to be post-Lake Bonneville, which would be less than 13,000 years old. And it's likely that they're a few thousand years old, maybe 3,000 to 5,000 years old, but we don't really know. And aging these crumbly carbonates is really difficult. I have a Green River Formation stromatolite here on my desk, and you can see the microbial mats form these nice arcs. I have another one here that maybe you can see a little better. And so these are not the normal angle of repose for sediment. And so you can tell that biology was here. And so that's what we see in the fossil record. These modern ones, we don't really know how old they are because the matrix is very crumbly and there's more sediment coming in and getting collected over time. And so they've been very difficult to age. That's what I'm hearing from geology friends, but likely 3,000 to 5,000 maybe years old. So that was one of the interesting questions. Other questions about, have we measured the pigment? Have we isolated the organisms? And we haven't. Melody's work was really about slicing up those things and getting the genes out, and not about trying to isolate organisms and grow them in the lab. Because then we would miss all of the organisms we couldn't isolate. So we haven't done that. And then Aaron had a really good question about pigments and other compounds that might be found in the microbiolite structures. And we haven't done that. We were just doing a genetic analysis. So those were some of the questions, Jaimi.
I have a question for Phil.
Mm-hmm.
Phil, sometimes I use the word egg and sometimes I use the word cyst. Is that appropriate, or am I going to be blacklisted?
Totally inappropriate, but we won't blacklist you. No, I think egg is very much more a colloquial term. I mean, people know what you're talking about with it, but in actuality, yeah, they're embryos, and so it just depends on the context. If you're going to tell somebody they're an encysted embryo and they're going to give you the same blank look anyone gives me when they ask what I do for a living, then call them an egg.
I know you're a sea monkey biologist, Phil.
I actually yelled at people all the time for using the term egg. And I thought I'm not I'm not an animal biologist, right? So I just thought, oh, cyst is the right word. Why can't you say cyst? It's not hard. I can teach kindergartners to say cyst, right? And then somebody said, well, it's an embryo inside a hard shell, right? And I'm like, yeah, well, why is that not an egg? I couldn't answer that, Phil.
And
no, I would love to provide a detailed definition for the exact differences, but I'm not sure I could do it justice, so.
well, and I came, the brine shrimp harvesters that I worked with, called them eggs. And so I use it, very interchangeably. And I think in my old brain, I can't even get out of it. So I'm trying to justify using egg and not cyst sometimes.
Yeah.
I just want to know from a real biologist, like, is it an egg? Is it the same thing as an egg, or is it different than an egg in some distinct way that we can? I want to ask a real biologist.
Wait.
Yeah, I should look up the exact definition of that. Because now I'm curious.
I mean, we can't be wearing brine shrimp scarves and not really know the answer to this question. It's bothersome.
I actually want to go back to like this primary productivity question. Why haven't we, this is all kind of very new. Why have we not done any primary productivity studies on Great Salt Lake? Do you have thoughts on that?
It was challenging. Back in the day, people did it by proxy. So they would measure chlorophyll in the system, and then they'd say chlorophyll equals this amount of productivity. They actually weren't measuring carbon fixation. They were looking for presence of chlorophyll, which they related to the number of algae species in a certain milliliter of water. And so that proxy measurement is what was historically done. And that's just not the way people measure it anymore, because we have better methods. So the better methods have not been applied to Great Salt Lake, and the proxy measurements were really done so long ago. And the paper itself by Stevens and Gillespie, they talk about all the trouble they had coming up with controls and normalizing the values. And they're like, we think it's this, but we're telling you why we're worried about our methods. So I was really struck by reading that recently and going through like all the problems they had in even using that method. And so the work that Eric Dunham is doing, and he can speak to that, is way more specific about watching where the carbon goes and getting an actual measurement of that. So it's a much better measurement. It's much more accurate. Do you want to say something about that, Eric?
I'm happy to. I'm going to have you repeat that a little bit. I was busy working on an answer to a question in the chat. So
that's okay. I was just saying that measuring carbon fixation is more accurate than measuring chlorophyll as a proxy. And the old studies were done by measuring chlorophyll.
Sure. I think, I've seen some literature really just in my, I've been kind of scrambling to get these calculations done for today's presentation. And I did see some literature questioning, the reliability of using chlorophyll to estimate biomass. And I think, as you said, the rates we measured are great, and they're going to be, I trust them to be accurate for the conditions of the lake. And I think the way that we've been able to collect chlorophyll data specific to our sample site, correlating chlorophyll to grams dry weight mat material, and then normalizing our carbon fixation rate data to grams dry weight mat material, I think we'll be able to get a decent answer specific to this system by using chlorophyll measurements. But yeah, it's always, you know, it does seem like a little bit of a dicey proposition to use chlorophyll as an estimate of biomass across varying environments.
Yeah.
I think it's appropriate for our system, since we're taking, you know, measurements of all of those parameters on each of our sampling trips. We should hopefully be able to, you know, to account for that variation.
And I think also, I mean, we're continuing to measure chlorophyll as we're doing this carbon fixation so that we can compare it backwards.
Exactly. Yeah, exactly. We'll be able to, you know, to normalize within our own data sets.
It has been shocking, though, how different the lake is every season.
Yeah.
I mean, particularly Phil and the brine shrimpers tell us this all the time. DWR tells us this all the time, but I've been kind of amazed by it.
Well, it's been fun for, you know, I was born in Salt Lake City and, you know, moved away when I was like 3. So I don't really, you know, I don't remember going to the lake when I was little. And so these sampling trips have been my first real exposures to it. And it's been a different experience each time.
Yes, yes, absolutely.
Do we have more questions? Are you nerding out, Matthew?
Oh my God, I have so many questions. I'm just waiting.
Do you want to ask some questions? Ask.
Well, I think, well, okay, so first of all, I think that this is such an amazing team that you guys put together for this conversation. Eric and Phil, I would love to sit down with you guys and just like do shots for biomass estimate methodology and stuff, because you've got chlorophyll, you've got Phil, you're doing net export of biomass from the lake with your brine shrimp cysts, and you can, so I think what you guys are really trying to understand is, you know, what is the net function of the lake for the ecosystem? And you're asking that in different ways. What's the standing crop of the biomass of the algae via grams per liter of dry weight of chlorophyll content? And then you're also looking at carbon fixation rates, okay? How much carbon is being taken up per unit time per volume? And it's fascinating how much data you guys have on this. So I'm thinking that with this team that's right here, you guys could get together, and your information, you guys could really, you know, back of the envelope, at least to start, find out what the real biomass is that's created by the lake and exported. And I think this is such a neat thing to be able to figure out, and then compare it to some of the more productive areas of the ocean systems as well. I mean, where are we on that? I mean, do you have like a mass per unit area? And then where does the Great Salt Lake fit in to other water bodies around the world? Sorry for rambling.
Oh no, that's fine. I mean, that is one of the most exciting parts of this project, and it's something that you know, we've been kind of going back and forth in this question and answer chat window about that. It's a tricky thing to come up with an accurate estimate for. There's been a paper, Stevens and Gillespie back in the 70s made the first, you know, took the first crack at it. And the calculations I made for today's presentation are, you know, several orders of magnitude lower than their initial calculations. And we, you know, we're not totally sure. I think, you know, that could be a fact. You know, they discuss in their paper how there are limitations to the ways they controlled their experiments and set them up, and you know, there's a lot to account for here. But there's also quite a bit of limitations in the calculations and assumptions I made for my presentation today. So I wouldn't be surprised if there's plenty of error on both sides, and we'll sort of see an answer converging in the middle. I think Stevens and Gillespie came up with something like 170 some grams of carbon per square meter of the South Arm
145.
per year.
145. You're close.
145. So grams of carbon per square meter of the South, you know, assimilated per square meter of the
grams of carbon per square meter per day?
Per year.
Per year.
And my calculations I made today work out to about 0.6 grams of carbon per square meter per year, which I think is low. And you know, this is the first time I've tried to do any sort of, you know, you're kind of, you know, you're taking a lot of external data, making a lot of assumptions, and propagating errors quite a bit whenever you do these calculations. So I mean, I have no doubt that my calculations for today are wrong to a certain degree. I kind of was able to compare them to productivity from the Great Lakes and from other aquatic environments, and they seem low to me. And I haven't had enough time with this data yet to figure out where those mistakes may be, but I wouldn't be surprised if my estimates come up, and I wouldn't be surprised if they're not exactly the same as what was calculated in the 70s too. So it'll be, I mean, that's what I'm most excited to do with this project, but it's gonna be tricky, especially when we try to take into account seasonal variation. As sunlight intensity changes throughout the year, and the time that sun is exposed to the lake changes throughout the year, and we have, there's a lot of variables to take into account, and I'm not sure that I have done all of that totally accurately so far. It's been a fun exercise, but I definitely have a lot to learn.
Quick question. So when you're talking about your in between point, what was it, 0.2 grams to 160 grams?
Yeah, something like that.
Per meter squared year. How does that compare to the export of just the cysts, Phil, that leave, that you guys harvest and ship out? I mean, if there's ever a real measurement of the carbon that leaves the system, that's one that's, actually weighed and regulated. So yeah. How do those two numbers jibe with each other?
I don't think we actually know how they jive with each other, and I'm not certain if we know exactly how much material carbon leaves the lake from the cyst harvest. It's something that could be figured out. People are considering, other researchers are considering this concept, but they're looking at it from, with even more complication, from one trophic level higher. The DWR Technical Advisory Group that has a scientific panel on it that we're part of, one of their avian researchers from Utah State University, Mike Conover, is formulating research questions currently about how much nutrients may leave the lake system through, the bird populations as they come through, and not just what they may take with them. Remember, millions of grebes coming, feeding, gaining biomass, taking that with them to their wintering grounds, but also how they might contribute to cycling of these materials while they're here too. So it's a great question, and it just gets more and more complicated the more we look. But there are people that are considering this type of topic, and so yeah, it just takes a while to get some of this information rolled out.
Yeah, exactly. Now, I did try to cross-check that today, but I didn't have time to look at, so my calculations are all just made for grams of carbon fixed per year. And, a cell or a cyst of a brine shrimp is not made of just carbon. So there's going to be additional accounting to be done there to account for, we have good measurements of the mass of brine shrimp cysts exported from the lake per year. But to figure out, the mass of cysts, how much carbon is in that number is going to be more work that I haven't had time to get to yet.
I'm sure it's on a nutrition label somewhere, wouldn't it be?
It might be.
Know how much saturated fat leaves the lake.
Yeah.
Hey, well, on that note, we've hit our 8 o'clock mark, and I want to thank everybody, especially our panelists, for coming in and telling us about all of your research. If you have kids at home, you might want to check out Mission Unstoppable. Dr. Bonnie Baxter is going to be talking about her wonderful pink microbes on Saturday morning at 9 a.m. That's Mountain Standard Time. But look, if you're not in Utah, check out CBS Mission Unstoppable and yell at us if you have any questions. You can always email me and I can send you links. And just thank you so much. And I wish we could nerd out more to brine shrimp, but we will just tell everybody good night with my little brine shrimp puppet. And I will email everybody links. Once I get this up on the internet, on YouTube, I will email you links. Good night.
All right. Thanks for hosting.
Thank you so much, Jaimi.